Hi, I'm Jessica Pruitt, and I'm currently a postdoc at the University of Mississippi working on an Embrace-funded project with the goal of integrating lab, field, and policy research to help inform sustainable oyster reef restoration efforts in the Mississippi Sound. And so before I begin, I just want to thank SURF for the opportunity to present this research as one of the anchor talks for this session. Now, as everyone at this conference probably knows and appreciates, oyster reefs are important ecosystems. Besides the fact that people like to eat oysters, they also form large biogenic structures that provide a habitat and nursery for other economically and ecologically important species found in estuaries. Oysters also provide other ecosystem services, such as water filtration and shoreline protection. Yet wild populations are in decline worldwide due to overharvesting, which has been exacerbated by habitat degradation, disease outbreak. Now, in the early 2000s, oyster catches in the Gulf of Mexico were some of the highest in the world, but they have declined dramatically in recent years. For example, in Mississippi, commercial landings were averaging about 400,000 sacks annually between 1998 and 2004, but this has been reduced to just 20,000 sacks annually between 2015 and 2019, and there was no harvesting allowed in the past 2 years. And these devastating losses have been due to the disasters in the Gulf, such as the 2010 Deepwater Horizon oil spill, multiple major hurricanes, and freshwater flooding events, such as those caused by multiple openings of the Bonnie Carey spillway. So these spillway openings divert freshwater from the Mississippi River into Lake Pontchartrain in Louisiana after periods of heavy rainfall in the Mississippi watershed to help control flooding in areas along the river. And then shown in this infographic in the right, the opening frequency and duration of the spillway has increased in recent years, with 8 openings in the past 15 years, if we add the 2020 opening. And it was opened 3 years in a row between 2018 and 2020, including twice in 2019. And so this freshwater introduced into Lake Pontchartrain then flows into the Mississippi Sound and the rest of the Gulf, introducing large amounts of fresh water into the estuarine waters that can reduce salinity levels to nearly zero, as well as increase nutrient and pollutant levels, which causes algal blooms, as shown by these colorful areas in the 2 satellite images on the left. And algal blooms are a problem because, one, they can be toxic in nature, such as micropersistent producing freshwater cyanobacterial blooms that were reported in 2019 during the spillway opening and caused widespread closure of Mississippi beaches, as well as the death and decay of algal blooms lower dissolved oxygen and pH levels in the water. We just have to look west of Lake Pontchartrain to appreciate the effects of eutrophication in the Gulf. So this graphic shows the frequency of bottom water hypoxia, which is when dissolved oxygen levels are less than 2 milligrams per liter, in which hypoxia is present nearly every year in areas in red, which are closest to the mouth of the Mississippi River. And so all these stressors associated with spillway openings have had detrimental impacts on oyster populations, as seen with a mass mortality of oysters in the western Mississippi Sound after the 2019 openings. Yet, on a positive note, across the world there have been a surge in oyster reef restoration projects, in particular in Mississippi. So in Mississippi, these efforts are largely involved deploying culture material in areas that have historically contained oysters in hopes that wild population of oyster larvae will settle and over time form a functional reef that can be harvested. Yet restoration success is dependent on their survival of these early oyster life stages, which are generally found to be less tolerant to environmental stressors than adults, but this is less well studied. And so the goal of our project is to assess how these early oyster life stages respond to water quality stressors, and particularly those associated with freshwater flooding events, like the Bonnie Carey spillway openings, which have decimated the adult populations in the Mississippi Sound. Then we aim to translate and share these findings so the best available science is actually available to research managers to help them inform their decision-making in oyster reef restoration efforts in coastal Mississippi. And then lastly, I'll briefly discuss some of the local partnerships that we've engaged in to help make this project possible. So for the science, we exposed 4 different early oyster life stages in the lab to either low dissolved oxygen, low pH, or low salinity in isolation or in combination. And we assessed the growth and survival, the early life stages, in response to these flood-associated stressors. So we also exposed the oysters to the 2-way combination of all these stressors, but just for the ease of showing you all the results in a short amount of time, I'm just going to show the single and 3-way data. So the next few slides are going to have graphs that look like this one, in which the oyster response will be on the y-axis. So in this case, larval growth, and the different stressor treatments, which is shown in the x-axis. So we found that when we exposed 48-hour-old veliger larvae to these stressors for 96 hours, there was reduced growth in the low DO and low salinity treatments, as well as the combination of all three stressors. And so these photos are representative of what the larvae looked like at the end of the experiment. So you can see that larvae exposed to all three stressors were smaller than the control and also had some sort of developmental deformities, it seems. Next, when we expose competent pelaviger larvae, so these are your larvae that are ready to settle on suitable substrate, we exposed them for 48 hours to these stressors, and we found that settlement was reduced when exposed to all three of these stressors, either independently or in combination. Next, when we exposed juveniles that had just settled 24 hours prior for 7 days to these stressors, we found an increased mortality in the presence of lower salinity, in which about 75% of the spat died in the low salinity and multi-stressor treatment. And then finally, we exposed 4-week-old juveniles that had been reared at the hatchery prior to the exposure. They were about 5 millimeter shell length, so that's smaller than my fingertip, as you can see in that photo. When we first began the exposure, so we exposed them for 24 days in the lab. Now we found that only seed exposed to all three stressors had increased mortality relative to the control. And this wasn't as high of levels as the day-old spats, so it's only about 15% mortality compared to that 75%. Yet we also saw reduced growth due to the stressor treatments, which we measured as the change in wet weight during the lab exposure. So there was a reduction in growth in the low pH and low salinity, and there was actually no change in weight when these seed were exposed to all three stressors. So if we put these results into context of the eastern oyster life cycle and oyster reef restoration efforts, if you deploy culch in hopes of creating a restored oyster reef in an area that's sort of prone to flooding disturbances or has suboptimal water quality, even if you have adults spawning in the area that's bringing in larvae, you're going to get reduced growth of these larvae, causing them to spend a longer time in the water column to reach a competent size to settle, at which point you're going to get reduced settlement onto your substrate, reducing recruitment to your attempts at a restored oyster reef. And then if you do get settlement on these reefs, you're going to see increased mortality, especially in this first week of settlement, further reducing recruitment. If you've got some juveniles that grow up a little bit, these growth rates are going to be slower, and this is going to cause a delay, at which point these oysters reach sexual maturity, ultimately reducing recruitment overall and creating this negative feedback loop, in which you're going to have probably a low probability of actually creating a successful restored oyster reef. And so the next step is to translate this science and make it readily available so it can be used by decision makers involved in oyster reef restoration efforts. So this part of the project is headed up by the lawyer on our team, Stephanie Otts, who's the director of the National Sea Grant Law Center, which is based at the University of Mississippi Law School. And so we're putting together a policy brief to share with the Mississippi Department of Environmental Quality and the Mississippi Department of Marine Resources, who are both in charge of oyster reef restoration planning in Mississippi. So if you're like the scientist on our team, what exactly is a policy brief? So it's a short, neutral summary about sort of what is known about a particular issue or problem. So in this case, oyster reef restoration, written for non-specialists. And there's sort of two types of policy briefs you can write. One that's arguing for a particular policy, or one that's just providing balanced information on a particular problem. So this is the type of policy brief we're putting together to share. Well, today I just presented the results from our lab experiment. The fieldwork for this project has relied heavily on partnerships from the coast, especially considering Ole Miss is about 5 hours away. So this includes the marine science interns at St. Stanislaus High School, located in Bay St. Louis. They've been a tremendous help with our field deployments of oysters and water quality monitoring sensors. We've also got to engage with them through a Lunch and Learn Zoom seminar series that we plan on doing again this school year. Additionally, the folks at the Mississippi Oyster Gardening Program have been generous enough to share some of their oysters with us. And so this work is actually the focus of JC's talk, which was yesterday, but you can listen to it again. So the Oyster Gardening Program is designed they provide spat on shell to local community members that have a dock, in which they'll sort of raise up this spat until they get to large enough age in which they can be outplanted onto restored oyster reefs. And so with that, I just want to thank so much the other members of my Embrace University of Mississippi team. I want to thank, again, Letha Boudreau and the interns at St. Stanislaus for all their help, as well as Scott Ricard at the Auburn University Shellfish Lab for providing all the oysters for this project. Thank you all so much for listening.